Commissionerate College Education, Vijayawada. I am Dr. K. Daniel, HOD of Zoology JKC College, Guntur, Andhra Pradesh. In this video lesson, I would like to explain about the DNA modifying enzymes, that is DNA polymerases. Let me show the contents of this lesson. The first one is introduction about the DNA polymerases. The second one is the properties of DNA polymerases. Third one is the process of DNA synthesis by DNA polymerase enzymes. The fourth heading is types and applications of DNA polymerases. Of course, the fifth and sixth points include self-assessment questions and web links. Now, the first one. What are the DNA polymerases? How can we define these DNA polymerase enzymes? The enzymes that add successive nucleotides to growing DNA strands are called polymerases. That means these DNA polymerases, they are useful in the elongation of the DNA double helical structure. All the living organisms have several DNA polymerases in their body cells. Every time a cell divides, DNA polymerases role is required to help in the duplication of the cells or cell's DNA, so that a copy of original DNA molecules can be inherited to each of the daughter cells. In this way, genetic information is transmitted from generation to generations. So, the DNA polymerases, they play a very important role in the inheritance or transmitting the characters from parents to offspring. The DNA polymerase enzyme was first isolated from a bacteria, a familiar bacteria, which is called Escherichia coli. Escherichia coli by Arthur Korenberg in 1976. Hence, this enzyme is also called Korenberg polymerase, as it is named after his discoverer. The second one, what are the properties of DNA polymerases? All DNA polymerases synthesize DNA in 5 to 3 prime direction, adding a DNTPS, means deoxynucleotide phosphates, to 3-hydroxyl group of the growing site. This is the first property. That is, the DNA polymerase synthesize or elongate the DNA molecule in the 5 prime to 3 prime direction, adding the nucleotide sequences in the hydroxyl group end, that is 3-hydroxyl group of the growing chain. And a second property, these enzymes simply add a new DNTPS or DNTPs to a performed primary strand that is hydrogen bonded to template. Third property, DNA polymerase enzymes not only add nucleotides, but they can also remove nucleotides either from 3 to 5' ends or from 5 to 3' ends of the DNA strand. That means they not only add the new nucleotide to the DNA sequence, they can also remove, or they can also take off the certain nucleotides from the DNA molecule. This activity, or the removing of the nucleotides from the DNA molecule is called exonuclease activity. It is called exonuclease activity. So this type of activity, that is the activity of removing of nucleotides from 3' to 5' end of the strand, is called proofreading activity. That means the exonuclease activity can be taken place in two ways. The first one is the removing of nucleotides from 3 to 5' end of the strand. This is called proofreading activity. This is called proofreading activity. This is done mainly to remove the mismatched nucleotides just synthesized. So there are certain nucleotides which are not matched with the already synthesized DNA molecules. So during such cases, these DNA polymerase, they can remove such mismatched nucleotides. So this is one type of exonuclease activity. The second type of removing of nucleotides is, it is nothing but the removing of the nucleotides from 5 to 3' end. That is to remove the attached small DNA fragments on the templates. These already attached small DNA fragments are called Okazaki fragments. What they are called? Okazaki fragments. So this is a second type of exonuclease activity, which is the functional activity of the DNA polymerase enzymes. And as far as the another important property, these DNA polymerases cannot initiate DNA synthesis de novo. That means they cannot initiate themselves. They require some what you called a type of nucleic acid, a short form of nucleic acid, which is called a primer. So primer is nothing but a short nucleoside and nucleic acid sequence which is to be required for DNA synthesis by the DNA polymerase. Okay, these primers are very important in DNA synthesis, without which the DNA polymerase cannot synthesize or elongate the DNA chain or DNA molecular chain. So this is another property of DNA polymerases. And as far as the structural property is concerned, this enzyme has 3 sites for attachment. 3 sites for attachment. The first one is the template site. Template site for attachment of the template or DNA template. The second one is the 5' triphosphate site for attachment of deoxynucleotide triphosphates, DNTPs. And third site is the primer site for attachment of 3'-hydroxyl end of the RNA primer. So generally the primer is a RNA. It is the short form of the nucleic acid sequence, which is useful for the elongation of the DNA chain. Here you can see these one or two properties of DNA polymerase, especially the proofreading. Proofreading means it is first exonuclease activity, where at the 5 to 3' end there are certain mismatch nucleotides which has to be taken off, which has to be removed from the growing DNA chain. Okay. And the third aspect regarding the DNA polymerases are the process of DNA synthesis. The process of DNA synthesis by DNA polymerases. How the process takes place during the DNA synthesis. And the first and foremost step In the process of DNA synthesis is the requirement or the presence of an enzyme which is called replicase. So the polymerase which replicates the DNA molecule is called replicase. Why it is required? DNA double helical structure is twined. Double helical structure means it is twined, intertwined each other. So to unzip or to unwind for such type of DNA molecule, this enzyme is required, which is called replicase. Replicase is the enzyme which can unwinds or unzips the double helical DNA molecule into two single strands of DNA that can be used as a template for replication. Each single strand DNA can be used as a template for replication. What is replication? It is nothing but duplication of the original DNA molecule. And the DNA polymerase now adds new free nucleotides to the 3' ends of the newly forming strand, elongating it in the 5 to 3' direction. So you remember that the elongation or the growing of the chain is from the 5' to 3' direction only. That's why it is a very important property through which the process of DNA synthesis can be taken place. As I told, as the DNA polymerase cannot begin the formation of new chain on its own or de novo means it cannot synthesize its own by its own. It requires a short DNA nucleic acid sequence, which is called a primer for the growth of the or elongation of the new DNA chain. Here you can see how the replication takes place, how the DNA polymerase create DNA molecules by assembling the nucleotides. Here you can see two strands, 3', 5', one strand where the replication fork in the center and 5' or 5' to 3' end. So here the DNA polymerase, how it acts from 5' to 3'. From 5' to 3'. This is the green colored, depicted green colored is the DNA polymerase. It acts in this direction only, 5' to 3' direction. Okay. So where the replication fork starts, from there, where the replication starts from 5 to 3' end, it is called lagging strand, whereas the second one is called leading strand. Okay. Now, fourth heading of this topic is the types of DNA polymerases and their replications. So, based on the living cells they present, DNA polymerases are of two categories. The first one is the prokaryotic DNA polymerases. That means, what is prokaryotic? Prokaryotes are nothing but the bacteria in which the nucleus is not distinct. There is no distinct nucleus in the bacterial cells, hence they are called prokaryotes. So, in the prokaryotes, the DNA polymerases are first discovered, and that's why first we learn about these prokaryotes or prokaryotic DNA polymerases. There are so many types, but there are three important types of DNA polymerases in prokaryotes. They are, number one, DNA polymerase 1. This is called poly-1. Number 2, DNA polymerase 2. And number 3, DNA polymerase 3. So let us see one by one quickly. This is the DNA polymerase 1. So one diagram made by the crystallography. And as far as this poly 1 or DNA polymerase 1 is concerned, this was first isolated by a scientist called Arthur Korenberg in Escherichia coli, the bacteria, in the year 1955. They were first isolated by Arthur Korenberg, and it is called Korenberg DNA polymerase. And it is having a single subunit with 109 kd. Kd means kilodaltons molecular weight. Single unit having 109 kd molecular weight. And this enzyme elongates the DNA chain in a manner that it adds 15 to 20 nucleotides per cell. It can add 15 to 20 nucleotides per cell. That is its activity. And it is the most abundant enzyme, accounting for more than 95%, more than 95% of other DNA polymerase enzymes. So this is a diagrammatic representation of polymerase 1, how you can see the leading strand, and this is leading strand and this is a lagging strand, where the DNA elongation takes place from here, or here it is from 3' ends to 5' end. So lagging strand and DNA, leading strand, and there are other enzymes, polymerase 1, 2, 3, DNA ligase, so on and so forth. This is the replication fork where the helicase enzyme acts upon. Okay. Next, what are the applications of this DNA polymerase 1? So the primary function of DNA poly 1 is polymerization. What is polymerization? It is nothing but the synthesis of DNA. So in the replication or synthesis of DNA, it acts, or this is the main function of this poly 1. And not only that, it also has a subsidiary role in repairing the DNA. It can also remove certain damaged nucleotides, okojokai fragments, and it is also involved in excision repair with the 3 to 5' or 5 to 3' exonuclease activity, as I told you already, and in processing of okojokai fragments generated during lagging strand synthesis. So these are the 2 or 3 important applications of DNA polymerase 1. Next, DNA polymerase 2. This enzyme was first isolated by Thomas Korenberg. He was the son of Arthur Korenberg, who discovered the DNA polymerase 1 from Escherichia coli itself. And as far as the structural aspect is concerned, it is also having single subunit with a 90 kilodaltons molecular weight. And as far as the applications of poly 2 enzyme is concerned, it can seal the okojokai fragments and carry out the function of the polymerase, DNA polymerase 1 in its absence. That means in the absence of DNA polymerase, it acts as a DNA polymerase 1, where it can remove the okojokai fragments, or it can also have the exonuclease activity. That is, it shows its involvement in DNA repair. And the third one, DNA polymerase 2, this is another type. The function is not yet known, but it was isolated from Thomas Korenberg and other scientists, that is A.M.L. Jefter in 1970 from E. coli bacteria. It has 3 important subunits. Here you can see this is one subunit, this is second one, this is, of course, each subunit can be divided into subdivisions, but of course it is comparable with the eukaryotic DNA polymerase. So the core consists of three subunits with a molecular weight of 900 kilodaltons. The main character or activity of this polymerase is it has only 3 to 5 dash exonuclease activity. It has only 3 to 5 dash exonuclease activity. And it requires auxiliary proteins for synthesis of long DNA chain. That means definitely in the presence of polymerase 2 or polymerase 1, it can act perfectly in the synthesis of DNA. And of course, these are how DNA polymerase 1 and 3 work like this. Here you can see the primase and here the DNA polymerase, how the elongation of chain and all these things are given here. Okay. And the second type of DNA polymerases are eukaryotic DNA polymerases. As I told you, based on the origin of cells, there are two types of DNA polymerases. The first one is the prokaryotic DNA polymerases. The second one is the eukaryotic DNA polymerases. So eukaryotes have 10 DNA polymerases, about 10 DNA polymerases, in which mammalian polymerases are distinguished by Greek letters. Greek letters. For example, alpha, beta, gamma, epsilon, tau, delta prime and delta, theta, chi, psi, respectively. So, the DNA polymerases are given the letters of the Greek suffixes. These enzymes use the single-stranded DNA chain as template and 4-deoxynucleotides as precursors for DNA synthesis. This is the property of eukaryotic DNA polymerase. So they can use the single-strand DNA chain as template and 4-deoxynucleotide, that is DCTP, DGTP, DATP, deoxy, adenosine triphosphate, deoxycytosine triphosphate, guanine triphosphate, like that, as precursors for DNA synthesis. Of course, this is the diagrammatic representation of eukaryote DNA polymerase. So, what is the main function of eukaryotic DNA polymerase? The main function of eukaryotic DNA polymerases are that they are involved in replication of both nuclear and mitochondrial DNA. So mitochondrial DNA is there now. So they are not only useful in the replication of the DNA which is present in the nucleus, but also the DNA present in the mitochondria. So that is about the eukaryotic DNA polymerase. So totally, at the last you can see the similarity of both prokaryotic and eukaryotic DNA polymerases and their work over here. So this is double helical structure. Here there is RNA primer to initiate the elongation, and helicase enzyme is there to fork this or to unzip the double helical structure into single-stranded templates, from where both the lagging strand and the leading strand take its action, and like that a replication takes place, elongation of the DNA chain takes place. So this is about the DNA modifying enzymes too, that is DNA polymerases. So here I am going to conclude this lesson. I have given here the self-assessment questions like essay questions, short questions for the examinations, and also you can refer the web links which I have given here. You can refer and you can gather some more information about the different types of polymerases. So there are different types of polymerases, not only DNA polymerase but also RNA polymerase, reverse transcript, so on and so forth. But I have given here only the DNA polymerase and its types and applications. Okay, learners, thank you very much for listening. Thank you, one and all.